Hi, I'm Susan Rudiger, and I'm the CEO and co-founder of the CMT Research Foundation. I'm joined today with Dr. Chris Lorson, who is a professor in the Department of Veterinary Pathobiology at University of Missouri. And his lab there focuses on translational efforts in neurodegeneration. But he's also the co-founder and chief scientific officer of Shift Pharmaceuticals, which is a biotech focused on delivering gene therapy approach for rare genetic diseases. Shift Pharmaceuticals had some early success in SMA, and now they're applying their technology to CMT with funding from the CMT Research Foundation. Thank you, Dr. Lorson, for joining me.
Thanks for having me, Susan. Appreciate it.
You're welcome. So tell me a little bit about your technology at Shift and why it's well positioned for CMT.
Well, Shift started about 2.5 years ago and was co-founded by Dr. Steve O'Connor and myself. And our primary focus was spinal muscular atrophy, or SMA. And with the success from Ionis in the development of Spinraza, it's really been shown that antisense oligonucleotides have become a new powerhouse for genetic diseases. And this is where we thought we could step in and perhaps make a difference with CMT. With CMT1A and PMP22, what we're trying to do is use an antisense oligo. And an antisense oligo is essentially a specific short synthetic sequence that targets a known sequence within the gene, or in this case, within the pre-messenger RNA. And the goal there is to kind of interfere with the way that the cell reads that pre-mRNA. And you can kind of think about it in terms of perhaps editing a book, where normally an editor would go chapter 1, 2, 3, 4, 5. What we're trying to do is interfere with PMP22 expression and create a misediting event where you get exon 2, 3, 5. And when that happens, the book no longer reads correctly. And so it's a way to interfere with gene expression, protein expression. And that's kind of one of the problems with CMT1A is that there's too much of this gene. And so we're trying to interfere with that process. And this is through this specific ASO-mediated interference of the PMP22 gene.
Okay, and so how is what Shift is doing a little bit different from some of the other ASO efforts out there?
Well, I think we're taking an approach where we're using kind of the intrinsic properties of the morpholino backbone, or sometimes referred to as PMOs. And what's interesting about these is that they've been in the clinic for years. So if you go back and look at Sarepta and Duchenne's, these drugs are remarkably inert. The amount that you can dose with is really pretty spectacular. And so what we think what we'll be able to do is find a specific candidate, one that we can drive expression in with a nice, robust dose, and get this effect that lasts. And so one of the things about morpholinos is that they last for quite a long period of time. So this isn't going to be an everyday kind of drug. It's ideally going to be on the months kind of time scale or longer.
Okay, great. But something that we would have to take as patients regularly, but maybe the intervals are much longer than a daily pill or shot.
Correct. I think that that's something that, if you were to say we have something that's quite effective, but you need to be administered this 3 times per year, and I'd say 2 to 4 times a year. This is really kind of a ballpark. I think that would be more amenable to something that is a daily occurrence, especially if it's going to be an ASO. And currently there are no orally administered ASOs. They're, at least minimally invasive with injections.
And one of the things we really like about our partnership with you is A, that you've had some early success with SMA, and B, that you're building off of a proof of concept. I mean, we've seen in previous work that when you target the PMP22 with an ASO, it can be effective in the animals, but now we've got to get that into humans. Can you talk a little bit about maybe the toxicity or the side effects that you would see in a standard ASO versus the kind that you all are developing at Shift?
Well, I do think the PMO backbone is particularly inert. So that's what I was talking about in terms of the ability to dose quite high. For whatever reason, that chemistry just doesn't elicit an immune response. It doesn't have significant toxicity or side effects. And so I think that's one of the really interesting features about this kind of chemistry is that they're going to there's always a trade-off, whether it's absorption, whether it's stability, whether it's cell penetration. But at the end of the day, the ability to develop chemistries that are safe is critical. And so I think when you're talking about the backbone of the PMO, that this is really something that can drive home a lot of therapies in the sense that, we've seen success in the clinic in other diseases. We know that patients tolerate these at levels that you could never dose any other kind of drug. And so I think the PMO backbone is really one that we're excited about, and I know that other companies are excited about. And so it's amenable to not just SMA, not just CMT, not DMD. These are things that you can apply to a broad range of genetic diseases. And so one of the things that we're doing that's a little bit different is that we're trying to interfere with the expression by modulating the splicing of this particular gene. So in other disease contexts, like in SMA, what you're trying to do is relieve repression. So you're trying to turn on a particular exon, which I think is intrinsically more difficult.
Okay.
Whereas what we're trying to do here is interfere or inhibit. And messing things up is always a lot easier than fixing things. I mean, I can see that, every single day.
Terrific. And so you're starting with CMT1A. Do you see this technology being applied to other types of CMT?
I think that it's probably more amenable to different types of any kind of disease where you're trying to reduce expression, and in particular from monogenic diseases. So if you're working with a single gene, you know what the target is. I think that's the kind of approach that is particularly amenable to an ASO strategy.
Great, great. So, and tell me where you guys are in the process right now. I know you've started the project. So, where are you in the process, and where do you hope that you'll be?
I think that's a really good question. I think with any drug development project, you've got a couple of questions from the get-go. One is delivery, the other is hit to lead. So you're trying to develop a particular series of lead candidates, and we're actually making progress on both of those fronts right now. So it's still early days, but we're encouraged. We've been doing well, I think, on both fronts, and so we still have work to do, but I think the targeting is really pretty intriguing, and I think that the ability to really interfere with particular regulatory sequences in PMP22 is really what we're most interested in. I think one of the things that's really cool is that this is leveraging what we've known in SMA and really moving it into a new area. CMT1A has no other therapeutic options. I think that when you walk into this kind of field and you say, wow, Shift could really make a difference, I think that every researcher kind of goes, that's what I got into this for. And so it's pretty cool to be able to wake up and say, if we could make a difference, that would be pretty profound. And the opportunity is there. And when you look at SMA, it's a rare disease, and it has multiple drugs that are approved and many more in the pipeline. Mm-hmm. A lot of people who sit on review panels might say, why do you need another one? How many rare diseases wouldn't go, holy cow, I'd love to have 3, 5, 7 different therapeutic options? Again, the first one's not always the best, you never know what's going to, it could be a combination. It could be that different patients, across the spectrum are going to respond differently. So I think that having kind of the opportunity to really make a difference in somebody's life is a big deal. And we're excited to meet that challenge.
Do you see with the progress that's being made that the patients become active and build this urgency and partner with organizations like Shift Pharmaceuticals to help get these things across the goal line?
I think if you look back 30 years, that things like this did not happen. And so it's become a completely different kind of landscape in the sense that they're very focused research foundations, like the CMT Research Foundation. Their sole mission, your sole mission, is to target CMT. And the amount of energy and drive that you can bring to that is really pretty remarkable. There are umbrella organizations which have been incredibly powerful over the years, and not to dissuade or discredit that. It's just that here's another layer or another level of research support and enthusiasm. So I think that both are important, and it's exciting to see how the landscape has changed over 25 years or so.
Yeah, it's been really fun to be a part of it too. And it's been fun to be a patient who's been able to work with experts like you and bring them into the CMT community and get you guys excited about CMT, which is a question that I have for you. Were you looking at CMT before you started talking with us? And what were your thoughts around it before we partnered together?
Well, one of those umbrella organizations happens to be the Muscular Dystrophy Association, or MDA, and they have been outstanding for just as long as a person can remember. And as a member of what used to be their scientific advisory committee and now research advisory committee, I've been reviewing grants for CMT for probably 13 or 14 years. And it always kind of struck a chord, and I saw the parallels that were really quite nice between SMA and CMT, which is probably why they kept putting me on CMT grants. And that was really, I kind of developed a personal interest in terms of, I like to review CMT grants. There are some colleagues at the University of Missouri who have been longstanding investigators in the CMT field, perhaps more on the fundamental or basic biology side. But I think like Dr. Michael Garcia at the University of Missouri, bringing another level of interest and then needing a translational partner. And so there were a number of opportunities that just kind of came together, and it really made a lot of sense. And then from the Shift perspective, it just kind of dovetailed with my growing interest.
Excellent. Excellent. So now that you have the funding from the Research Foundation, you can commit to this project for CMT. Are you looking at any follow-on funding for CMT at this point, or do you have some proofs of concept to do first?
I think that within the next 6 months or so, the idea would be to kind of leverage what we've done and move into NIH funding. Mm-hmm. And I think the best one would probably be an SBIR to start with.
Excellent, excellent. And that's good to know. I love being able to talk to the donor community and tell them that a small investment from the Research Foundation, and it's a fairly small investment that we have with Shift, could blow up into funding from NIH, which could blow up into more funding, which could then blow up into funding from Big Pharma. And that our little bit of funding can seed an idea and help you really get this proof of concept off the ground. Right now, we're funded primarily by the patient community. And these are patients who have come together to say, enough to the fact that we don't have any treatments for CMT. And I want to be a part of the solution. And I think it can be really intimidating when you hear the price tag for a neurodegenerative disease is $750 million to develop it. And you think, how many car washes do you have to get to 750 million? And what you're talking about, your vision with Fight SMA and our vision with the CMT Research Foundation is not that many, because our job isn't all 750. It's not the multi-millions. It's a little bit of seed money here and there that become catalysts for these ideas.
Right. And I think that, and you have to partner along the way, you know, whether it was the initial, and when you look at the SMA drugs, they were partnered with the research community, the research foundations. They used those to move into NIH funding, and then they started companies, and then those small companies partnered with larger companies, and then those mid-sized partners with, you know, the behemoths. And that's just kind of the way things work.
Yeah. So when you close your eyes and think about this PMO technology from Shift for CMT, what do you see in 12 months, in 5 years? What do you envision?
Well, I see a couple of different things. You know, I see a number of things moving forward in terms of research development. I see things moving forward on the clinical side. And the objective would be to have a suite of clinical candidates that are progressing at various stages. And I think that's really the most exciting thing, is that you're able to grow from kind of a base, and you know what you're good at, and you can do that. You do this and you build out, you use your experience, and then you kind of see what it can be applied to. And I think SMA to CMT is a very logical extension.
Excellent. Thank you for saying that, because as a woman living with CMT, I've witnessed what's been going on in the SMA communities. In fact, I spoke with Jill Jarecki from Cure SMA a couple months ago, and we were talking about the amazing success that they've had as a community, driven a lot by patients, but also by all the partners. And I feel like it's CMT's turn. And I feel like we can have a lot of learning from what has happened with SMA, and building on a lot of the progress that basic researchers and basic research grants have delivered. We're building on that to get to the point that we're ready for clinical trials for CMT in a large scale.
I think that when you look at your website as well, and you realize that there is actually a portfolio of development, that there's a lot going on. And I think that's really important for rare diseases, is that you're able to generate the interest, that you're able to raise the dollars. And sometimes the right things don't get done, you know, at the federal level. And so raising awareness and really making it justifying further investment. And sometimes that investment requires really hammering home, hey, we're ready. And being able to be ready sometimes takes the, you know, foundations and the patient community to push it forward. So, and not that NIH isn't doing it, but sometimes, you know, it can be a priority even more so after a demonstration.
Right, right. And it's through partnerships with NIH, and through partnerships with FDA, and through partnerships with biotechs and academics and big pharma that it all happens.
Yeah, and when folks tend to work in a more collaborative environment, um, and in SMA, it was really pretty amazing to see the collaborative nature of things. You get a lot, you can push things forward that you wouldn't otherwise be able to do. And certainly you have to do what's right for you, but as a general community, being open and being, trying to push things forward for the betterment of the disease is really an important step.
Yeah, yeah. Because as patients, we don't have time to waste, and we don't have money to waste. And so collaboration helps expedite both of those things.
Agreed.
Yeah, yeah. Well, thank you for your time. I really appreciate it. And I look forward to working with you as this project continues. And I look forward to keeping our patient community informed about the wonderful advancements we'll make together.
Well, thanks again, Susan. Really appreciate the invitation.